Thank you. I'm delighted to be here back at Brunel University, a place that I remember incredibly fondly. And as mentioned, I went on to do my PhD looking at the cellular basis of learning and memory, what actually happens to the architecture in the brain as we learn and remember. I'm very much aware of the fact that you've all had lunch now, so you're probably feeling quite relaxed and sated. And so to kickstart this session, what I'm going to do is ask you to sit comfortably and stare at the center of this spiral. So keep your gaze there. You can blink, but do not, under any circumstances, look away. And whilst you continue to do that, I'd like to tell you a little story. All the way back in 1834, a Scottish rambler called Mr. Robert Adams decided to take himself out for an afternoon stroll. And whilst he was walking around the Highlands, he stumbled down into a little valley. And in the valley, he stumbled across a beautiful, mesmerizing waterfall. He was hypnotized by the downward cascade of water. He was enchanted by its flow, and he spent some minutes looking at it. When he finally managed to divert his gaze, when he finally managed to look away, his entire world had changed. In much the same way as when you all now look at my face, you should see something rather peculiar. It's a temporary visual illusion, so my face should go back to the right shape and size very soon. Hopefully that worked for the majority of you. What was happening there is a type of visual illusion that was first documented almost 200 years ago. But it's only recently that neuroscientists have been able to peer into the brain using this revolution in technologies that allows us to look into the brain with high resolution and in real time as a conscious, feeling, thinking mammal goes about its space and navigates its world and makes decisions. And using this new technology, we're really starting to get to grips with understanding the very nuts and bolts of our behavior and what makes us all us. So bear with me for a couple of minutes of brain theory now. I'm aware that you're all from kind of quite a broad spectrum of different disciplines, so I'm going to get down to basics. Our brains are made up of about 86 billion nerve cells. 86 billion, that's a huge number. It's about 14 times the number of people on this planet. To put it on a scale that is easier to comprehend, if we dug into your brain and took a piece of brain tissue about the size of a sugar grain, it would contain about 10,000 nerve cells. And each one of these 86 billion nerve cells that makes up your brain is composed of a cell body, a little blob, and extending out of that blob-like structure, the blob contains the nucleus, which is the director for the cell, if you like. And extending out from that blob is an axon, a long cylindrical structure that looks a little bit like a tree trunk, if you like. So if you put it the other way up, actually. So the tree trunk is extending up, and then there's the cell body, and extending out of that cell body in the other directions are arbors. They're called dendritic arbors, which are a little bit like the arborizations in a tree, the tree canopy. And the axon, this long cylindrical structure, is studded with sodium and potassium ions. And your brain uses about 20% of your daily energy quota, which is a huge amount. And the reason that it uses all this energy is to pump sodium and potassium ions in and out of those pores, those channels, in order to cause the zip of electricity, the passage of charge, to zip along that axon. When it reaches the end of that axon, it discharges as a chemical, and that electrically activates the next nerve cell in the circuit. And each one of your 86 billion nerve cells in your brain can be connected to up to 10,000 other nerve cells, which forms the most complicated and intricate electric circuit board with probably around 100 trillion connections within your mind. And your circuit board, this 100 trillion connection circuit board that makes up your mind, moves and changes all the time based on your experience through life. So each one of you has a very individual circuit board. The cartography of your mind is very different from the person in the seat next to you. Why is this? Well, because every single time we learn something new, we can make a new connection within our mind. And this is what I studied during my PhD when I went on from Brunel to Cambridge. I was looking at how new connections can form in the brain as we learn and remember, and how that affects our perception of the world, our sense of reality, and why this sometimes goes wrong in people that have distorted realities, for example, people that suffer from psychosis. So as we learn something new, a little connection forms from one of the tips of the arbors, the tree dendritic branches of the nerve cell. And this little worm-like structure reaches out from the tip of the tree canopy, if you like, in your nerve cell. It's a little worm-like structure, and it reaches out, it follows little cues from a neighboring nerve cell. Reaches out, forms a connection, and as you consolidate that learned thing into a memory, new proteins get recruited, so that this little Philopodia worm-like structure becomes more of a mushroom-shaped dendritic spine. And it literally is mushroom-shaped. It's studded with lots of receptors that allow that connectivity to communicate the electric signal from one nerve cell to another nerve cell. And then there's something quite beautiful that happens. It's almost like a Valentine's Day kind of romantic dance that can happen in your mind. If you repeatedly revisit that connection and make new associations in new ways with that new memory, then that stable memory connection in your brain can actually split into, it forms almost a heart-shaped structure as it's transiently changing. It splits from a heart shape, from mushroom to heart-shaped, and then forms two daughter spines, so two daughter connections, which give rise to two different routes within the brain in order to help you think in different ways. And what we're viewing here in this movie is, so these new technologies are allowing us to watch mammals as they move around the world and move around laboratory units. We can watch using really high-powered microscopy and watch as they learn to navigate a maze and go in a particular direction in order to find treats and then remember which way they need to go to get the next treat. What we're seeing here is if we tag certain proteins in the brain that are involved in helping that plasticity, these connections form in the brain that are involved in learning and memory. If we tag the proteins that are essential for that plastic process with green fluorescent protein, a dye, then what we're seeing here is the shuttling of these proteins that are essential for memories being made in the brain. So what we're watching is almost memories being made in the brain, and I find that incredible. So this is a video that's on loop. It's 15 minutes of video that's being made on loop. Now, there's something really key about the way our brain uses information based on its prior experiences. And I mentioned earlier that our brains use about 20% of our daily energy quota, which is a huge amount. And our brains don't want to just process all of the information that's coming in through our senses all the time, because that would be too exhausting for it. You might imagine that the brain would almost blow a fuse if that was the case. So instead, it filters the information that's coming in based on its prior experiences. It makes assumptions based on what we've learned before, and it has to do this in order for it to not explode, if you like. And there's a neat illusion here that really illustrates this point. So all of us are used to seeing faces rather than the back end of a hollow mask. And so our brains see this circulating mask. And when it gets to the back end of the mask, even though the shadow cues are telling us that the eyes and the nose and the lips are pointing backwards, our brains ignore that information that the shadows are giving us. And instead, we just overlay the assumption that we presume another face is popping out. There's a more extreme version of this here. So this is the back end of the mask flipping round, and you're just ignoring those shadow cues and instead seeing the front end of a mask on both sides. And no matter how much you tell your brain to see the back end of a mask, it can be quite tricky to do that. There's another illusion, an audio version of this illusion. I'm going to ask you to try and make sense of this file. pretty much gobbledygook, right? So the majority of you can't really make much sense out of that. So maybe you can decipher 1 or 2 words, but it's pretty much gobbledygook. Now try and listen to this audio file.
The camel was kept in a cage at the zoo. The camel was kept in a cage.
Poor camel. That's not very nice, is it? If we now go back to the original audio file, the gobbledygook, we'll see if your brain can understand it now. So very clearly, we're hearing that the camel was being kept in the cage at the zoo because the 2 audio files had a very similar cadence, and your brain was using that sense of the camel being kept at the cage at the zoo and overlaying that sense onto the gobbledygook in order to try and make sense of it. And some scientists like to think of the brain as this prediction machine. It likes to use prior information from previous days to try and predict and make assumptions about what's going to happen next. And this propensity within us, that's inbuilt within us, in our brains, in order to use its energy efficiently, unfortunately can give rise to people having cognitive biases, seeing things in particular ways based on their past experiences. And in fact, you can also think of the fact that each one of us has had some common shared experiences throughout our lives, for example, seeing faces rather than the back end of a mask. But we've also had some very unique and individual experiences in our lives. And the culmination of those 2 different aspects of our past experiences come together to give us a very unique cartography, connectome, the way that our nerve cells are connected within our mind. And it's this individual connectome, this individual map within our mind, these individual dendritic spines, how they form within this intricate space within our skull, that gives us our unique perception of the world and our unique sense of reality, and in turn instructs us how we should interact with the world, the type of decisions and choices that we should make through our lives. So that's a background basically into what happens to the brain as we learn and remember, and some of the interesting ideas and research that are going on around it. There's also some research and this phenomena, I don't know whether this is happening at Brunel University, but at Cambridge University, it's almost something along the lines of 1 in 5 students now are admitting to taking smart drugs or cognitive enhancers, in the hope that they might help them with their exam revision. So these smart drugs or cognitive enhancers were once thought to be in the realm of blockbuster films, of science fiction. But increasingly, students seem to be buying over the internet, so it's not strictly illegal, it's a bit of a grey area of the law, but they're buying the drugs that are traditionally prescribed for attention deficit hyperactivity disorder or Alzheimer's, in order to help these students. The students think that it will help with their focus and their attention and their memory. So there's not a huge amount of research in terms of how these drugs exactly work and whether they will really help the students to get a boost in their grade marks. What we do know is that it seems as though these drugs help focus the particular circuits within the brain so that only certain circuits within the brain are electrically active at particular points. So within your brain, there are these excitatory signals, so these kind of full-on 120 mile an hour electric signals that whoosh around the connectome really fast. There's these green go light signals, but there's also inhibitory red light stop signals. And it seems as though some of these smart drugs actually help to press the brakes and cause a red light stop signal to go on in other areas of the brain so that you can focus and concentrate on particular tasks without interfering information coming in. There's other ways that scientists have also been looking at enhancing brain connectivity, if you like. So there's been increased interest in the use of psychedelics for brain functioning. And I don't know whether you've heard recently of, in Silicon Valley, quite a few coders And actually, I read recently on the BBC that quite a few mothers that are going on play dates are taking small amounts, they're microdosing LSD. And they think that these small amounts of LSD that are taken once every few days helps boost creativity, to help the coders to think in new lateral ways in order to problem solve any coding issues that they might have, and also for the mothers to help them to play with their children in more creative ways. I'm not quite sure what I think about that, the latter one. I just read it a few days ago and I was like, oh, okay. Yeah, so scientists have been really interested in what these psychedelics, LSD for example, and psilocybin actually do to the mind. So they've been taking some volunteers and giving them some very small doses of psychedelics and then putting them in a brain fMRI scanner. So an fMRI scanner measures oxygen levels that are going to different areas of the brain, and it photographs the brain whilst measuring these oxygen levels. And oxygen is a good indicator of which areas of the brain are being highly metabolically active, because in order to use lots of energy, you need oxygen for the reactions to take place. So they put the volunteers in a scanner, give them small amounts of LSD or placebo, and then see what happens to their brain as they're asked to think in different ways. And what they're finding is that small amounts of psychedelics are literally lighting up much vaster areas of the whole connectome. They're engaging whole areas of the connectome. So it's working in a, if you like, in an opposite way to the way that smart drugs or cognitive enhancers do. So on the right-hand side here is a volunteer that's been given just the placebo, saline solution, and on the left-hand side here is a volunteer that's been given the LSD, a very small amount of LSD, and you can see large areas lighting up and the bright red indicating light. When you then take that information from the brain scanner and try and map it into the way that the different areas of the brain are connected together in this visualization way, then again you're seeing that there's a much greater amount of connectivity between disparate areas of the brain on small doses of LSD. And scientists are quite excited about this because they think that there's the potential for using these psychedelics to help people to think in new ways, not just to creatively problem solve, but for example, if people are suffering from ingrained addiction problems, or they are suffering from really acute or chronic depression, and they're kind of stuck in a negative way of looking at the world, and they're finding it very difficult to get out of that dark, ruminating place, then actually the use of psychedelics, and also people with post-traumatic stress disorder, for example, that may have come back from Afghanistan and experiencing the world in a very negative way, very small amounts of psychedelics seem to be allowing the circuitry of the brain to revert to a more naive, childlike state. So this brain where it's lit up in lots of ways without all that inhibitory action going on, the red lights in the brain aren't working. So instead there's excitatory activity going on across the brain, accessing different areas of the brain. This actually resembles much more of a naive, childlike state in the brain, which is why you sometimes think of children as being kind of quite tangential and creative, rather than focused and paying attention in one particular way. So there's been lots of preclinical tests looking at the use of LSD for people that are suffering from disorders, for example, depression and addiction or post-traumatic stress disorder. Now, I am by no means advocating that we should all go out and take small doses of LSD, or that we should go onto the internet and buy ourselves a whole range of smart drugs or nootropics. I'm not advocating that at all. And in fact, I specifically don't think it's a good idea for students to take to try any of these drugs. There's this amazing professor at University College London called Sarah Jane Blakemore, and she's created a whole new field of neuroscience study. And it's this idea, this appreciation that there's a very distinct phase of development within the brain, which is the adolescent period, which extends all the way from around 12 years old to the mid-20s. And it's this phase of brain development where there's a huge amount of plasticity going on. So if we think of our brain changes, typical brain changes during the lifespan, as a baby in the womb, as the brain is being formed, the neural circuits are being laid down under direction of the DNA in our genes and in the nucleus of our cell. That directs how these nerve cells' axons should extend out and how they should start to connect in the first place, to give us our crude map way by which we think. And then as we emerge as a baby from the womb, our brain's nerve cells are there. So all of the nerve cells really are there, but the nerve cells then continue with their refinement in connecting up with each other. And the brain actually grows by at least 25% in the first few years as those nerve cells extend out and form that canopy of branches and connections within the mind. Now, that process of connecting up continues to take place throughout our life, and we continue to hold this ability for plasticity to be able to form those connections in the mind throughout our life. But it's at its greatest rate during those first few years and in the womb. And then it was kind of thought that your brain just trundled on throughout the rest of your life, from when you were 10 years old. It kind of just trundled on, and then eventually, it withered and died. But there was some wonderful research that was done in the late 1990s by a professor called Rusty Gage that was at Stork University in America. And he discovered that there's something called neurogenesis, which is the birth of new brain cells, which can occur not just in the baby in the womb, but also occurs throughout our life. And there's key areas of the brain where this neurogenesis, this birth of new brain cells, occurs. And one of the areas that it occurs at its greatest rate is in an area of the brain called the hippocampus, which is a really small little area that's buried deep in the middle of the brain, and it's shaped like a kidney bean or a little seahorse. And there's a little area called the dentate gyrus within the hippocampus where new nerve cells can be born throughout life. And I'm kind of, so throughout our life, new nerve cells can be born. But there is a distinct phase, I'm going back to adolescence, so there's a distinct phase in adolescence when there's this neurogenesis that is still taking place. But on top of this birth of new nerve cells, there's huge amounts of connections that are being made. But layered on top of that, there's a fundamental phase where synaptic pruning happens. And this synaptic pruning is basically like pruning a bush into a particular shape. It's sculpting the brain canopy, the brain kind of branches, if you like, into a particular shape in the brain. And there's huge amounts of this that is happening during the adolescent years. And in fact, in the prefrontal cortex, the region behind your brain, behind your forehead, it's a region that's involved in flexibility of thinking, planning ahead, and reasoning. There's a whopping 17% decrease in the amount of connections that occur in the teenage brain. And that sounds like a staggeringly negative loss of brain tissue until you realize that actually that tissue loss is being replaced by fat wrapping itself around the axon, that long cylindrical structure in the nerve cell. So in effect, what's happening during the adolescent period, and as I said, this period, this neurodevelopmental period, extends all the way into the mid-20s, is that the brain is being sculpted so that it's losing a lot of its scenic roots in the mind, if you like. And those scenic root ways, these scenic ways of navigating thought, are instead being replaced by more high, kind of almost like motorways in the mind, because the fat wrapping itself around helps those electric signals to be conveyed from one nerve cell to another nerve cell at high speed and with high integrity. So really, the adolescent period is critical in terms of the individual finding their unique identity. They're losing different ways of thinking and replacing that variety in the way of thinking with more streamlined kind of habitual thoughts. That said, there's also an extra drive within adolescence. There's particular ways that the genes are expressed in the adolescent brain, particularly in the reward circuit of their brain. That means that they are more likely to be impulsive and be motivated to take risks and to be sensation-seeking. And this is kind of a stereotypical teenage behavior. But it's something that has good reason for why it is as well, because the formative years of that individual's life has really been shaped by, generally speaking, what their primary caregivers, so probably their mum and dad, the environment that's been created by their mum and dad. And during the teenage years, it's a great time because there's all this dynamic stuff, this pruning plus this plasticity that's occurring in the brain. It's a great time that the teenager can really start to find its way in the world and think in new ways. So it makes sense that it should enhance its experience repertoire by going out and seeking new sensations and seeing new environments and meeting new people and having different discussions during that life. But unfortunately, they may also be drawn, because of their neurobiology during this critical phase, they may also be drawn to seeking new sensations in terms of drugs of abuse, whether it's psychedelics or it's smart drugs. So I'm not advocating that the students do that. So are there any natural ways to boost brain power other than taking psychedelics and smart drugs? Well, number one is to stay physically active. That helps. There's been some wonderful studies that have shown that simply the act of running on a running wheel can enhance the rate at which neurogenesis happens. It can enhance the rate at which new nerve cells can be born in that hippocampus area of the brain, the region that's involved, heavily implicated in learning and memory. Staying socially active. So again, there's been some lovely studies that have shown that if you run, and also, so these initial studies were first done in mice, and there were little mice on a running wheel. And the mice that had free access to a running wheel and made use of it, and then also had a wonderful, beautiful cage that had multi-layers, and they had lots of friends that they could go and meet, and went around sniffing them and kind of conversing with them, if you like, and exploring different environments. Those mice had a much higher rate that these new nerve cells could successfully integrate within the existing circuit of their mind, and so their brain could flourish because they had this full access to this circuitry, and they were able to start to think in new ways because they had this greater scope for plasticity. And the same seems to hold true for humans as well, all the way into adulthood. If we stay physically active and socially active and explore new environments and new ideas, then that helps to boost the plasticity within our brains. Another piece of advice for brain plasticity, particularly in relation to learning and memory, is to check your diet. Generally speaking, anything that's good for the heart is going to be good for the brain. And part of the reason for that is that your brain requires vast amounts of blood carrying oxygen to get to it via the meninges that kind of cover the brain. And because your brain requires 20% of your daily energy quota, it requires vast amounts of that oxygen. So anything that helps your heart to pump that oxygen and that blood around is going to be good for your brain. Getting a good night's sleep seems to be incredibly good for brain health. Not only does sleep provide the opportunity for your brain to clear away any toxins that have built up within the brain during the day, but it also helps the process by which these little worms of learning from the day before can be consolidated into a mushroom-shaped memory spine. So it helps that process of memory consolidation. Neuroscientists who are working in higher education in particular always seem to advocate the benefits of continued learning throughout life. Again, it seems to help to encourage the plasticity within the brain. And also staying positive. It's very difficult to stay physically active or socially active or to eat well if you're feeling depressed. And so one thing that I do is keep a gratitude journal, and I try and think about the things that I'm grateful for at the end of each day in order to try and stay positive. If you're interested in any of this, then I've got a couple of books that are out, as was mentioned. There's a book on the science of fate that was launched last week, and also a book called Consciousness that was published with the publisher's Penguin. And if you've got any questions, then I'm going to take some questions now. Hopefully this will have brought about quite a few questions with it. But I also want to end by asking you to listen to this beautiful audio file. So I've kind of been giving you a whiz whistle stop tour through the brain incredibly quickly and focusing on learning and memory and how our brain builds assumption into its kind of mechanistic approach for how it operates. But obviously our brain isn't just there for us to learn and to remember and for us to interact with the world. We're also emotional beings. We feel things. And maybe part of the way that our brain is coded is so that we can feel these emotions to help keep us motivated to do things in the next days. But we're not the only species that do this. And this is a lovely sound of a mouse that is giggling. It's been taught to associate, it's learnt from its environment that if a hand comes towards it, and if it, in particular its kind of carer's hand comes towards it, looking like it's about to tickle it, then the mouse will anticipate this tickle and start giggling in much the same way as if you start to try and tickle a toddler, it will start squirming and giggling in anticipation. So this mouse has learnt that a hand approaching like this means that it's about to be tickled, and so it will start laughing in anticipation. And this is the noise of the mouse laughing. The sound has been transduced so that the human ear can hear it. And that's it. I'm going to end on that. Thank you. I've got lots of time for questions, haven't I?
Lots of time. That's great. Thank you very much, Hannah. I think for some time now I've wanted staff here at Brunel to have a bit of an introduction to neuroscience, because I think it's really important in terms of understanding how our students learn and also understanding their behaviour as well. So that was really helpful and I think really insightful for everybody. So Hannah's got lots of time for questions. There are some mics going around. So please, hands in the usual way, and yep.
Yes.
Two there.
Teenagers, if they start taking this risk-taking behaviour, presumably it will settle down as they get older. What is the difference between normal development and adult ADHD? They still continue to have that risk behaviour.
Mm-hmm. So in terms of, so I went recently to King's College London and I was watching, they've developed this new technology thanks to an EU grant that allows us to look at the connectome, the way that these nerve cells are put together. They've developed this new technology that allows us to watch that process occurring in for an embryo, a baby in the womb, from 20 weeks gestation. And this is an incredible feat of methodology, really, because the baby's obviously moving around and being bathed in this amniotic fluid. And what they're seeing is that those initial stages of the connectome taking shape, there's a particular way in which that is occurring that can predict whether somebody might go on to develop attention deficit hyperactivity disorder. And there does seem to be a high genetic basis for ADHD as well. Now, as I said, during the adolescent periods, there is this kind of biological, chronological switch, which means that the majority of adolescents will have this sensation-seeking behavior and be slightly impulsive. And there's very good reason for that. But within our population as well, there's a subsection who have an extra genetic disposition to be that way, to be sensation-seeking and to be impulsive and to enjoy novelty and adventuring. And there's real positives of that. So, for example, quite a lot of entrepreneurs have this particular genetic variant, if you like. It can be harnessed in a positive way, and it can also give rise to diagnosis of ADHD, which can be very difficult for individuals to manage, particularly in certain environments. So there does seem to be a genetic and connectomic link for ADHD, which is, if you like, it's at the extreme of the variation for the way that our brain can be built. Does that answer your question?
Yeah, that's great, thank you.
There was another question here.
Yeah. Do you define learning as a new connection or strengthening all the existing connections?
Okay, yeah, so I basically went through this in a really quick way as a whistle-stop tour because I was hoping to get lots of questions. But so, as you learn something new, what's thought is that generally speaking, you have this new connection forming. And then so this is this new connection, little worm coming out from one nerve cell to another nerve cell. As you then consolidate that learnt thing into a memory, it changes shape from this flexible worm-like structure into a more mature connection, which is much more robust and has the proteins and the receptors in its head that allows the processing of the signals to happen in a much more efficient way. And that is a stable memory connection. Now, the way that your nerve cells can connect together, and I was saying that there's about 100 trillion of these connections that occur in the mind, it's thought that you can make associations from nerve cells. So it's thought that the initial learning happens in the hippocampus, which is the area of the brain that's shaped like a seahorse, which is buried deep. And then, so that's the initial connectivity that happens. And then that extends out, it's got projections. There's nerve cells that then extend out and connect with the prefrontal cortex here, which is involved in decision-making and reasoning and kind of flexibility of thought. And then from there can also be connections that go into other emotional centers of the brain as well. So it all connects together to give you these associations based on what you've learned, if that makes sense. So one thing might not be based on one connection. It might be a web of different connections within the brain. And actually, what probably makes sense is that you try to embed anything that you've learned into a whole framework of experience. So for example, if a student is revising for an exam, if they try and remember the information that they're learning and they tag it with if they turn it into a song, for example, or a dance, and so they think of it in a melodic way, then it might trigger responses in the auditory cortex, which is involved in kind of sound processing, but it also might trigger more emotional responses as well because they're having fun dancing with their kind of fellow students and making up this song, if that makes sense. It helps to embed that memory, that information. So the more embedded the information can be within your framework of experience, the more it can be accessed, and the longer it will probably stay.
Ian, at the back.
This morning I asked, I talked about fun in the lecture theatre, and like a very bad scientist, I said that having fun improves learning. But I don't actually know if that's true or not. So that's my question. Does fun improve learning, or is it just perception of learning improves because you're having fun?
Probably the perception of learning is improved because you're having fun, but also, no, most definitely. And that's why at Cambridge University there's been a professor of play that's been instigated in the Faculty of Education there. It's been funded by Lego, who are really into, obviously they quite like people to play. They make money from it. But no, so the more enjoyment that you get from anything in life, the more motivation you have to continue to do it. And the reward circuit within the brain is involved in this area called the VTA that runs from the VTA, which is kind of at the base of the brain, that goes to the nucleus accumbens, which is a kind of P-shaped structure, which is quite small in the brain, that's just full of dopamine receptors and releases dopamine. And then that branches out from the nucleus accumbens all the way to the prefrontal cortex, and there's some lovely kind of connections there. But basically anything that really causes this nucleus accumbens to release lots of dopamine makes you feel motivated and makes you feel happy and makes you want to make more connections and to continue learning. And then to embed that information within your prefrontal cortex because of the projections, if that makes sense.
Really, really nice talk. I just wanted to get your opinion on the role of the gut microbiome in the brain development and brain function, because it's an area that's got a lot of hype associated with it. A lot of studies, I suppose, coming out are very much correlation rather than true effect. So I suppose, what do you think the influence the gut microbiome could be having on the brain, and can in turn that be manipulated? Is it strong enough that we could manipulate the gut microbiome to improve brain function, or as you said, people with brain diseases and stuff like that, can we potentially reverse that using the microbiome as a tool?
Yeah, I mean, lots of neuroscientists over the last 7 years have been getting increasingly excited about this thing that they called the brain-gut axis. And the way that, I mean, the ancient Greeks used to say that our gut was our second kind of brain, didn't they? So it's kind of, this idea has been around for quite a long time. But there's been more and more scientific studies on this. And there seems to be quite a lot of robust research that's occurring in this area. There's lots of nerve cells, loads and loads of nerve cells within the stomach. And these nerve cells can release things like dopamine that I was talking about earlier, that's involved in reward and pleasure, for example, and send signals up to the brain. And I think one thing to be wary of is that quite a lot of the initial highly publicized studies that were done in this area were actually funded by Danone, who do the little yogurt drinks. But that said, a lot of neuroscientists have been really interested in this research area, and we're starting to get some really interesting results. And I think at the moment, because we're living in a landscape and an environment where it's easy for us to get access to food, but food that's not very nutrient rich, that might be affecting our microflora and the way that our stomach sends signals to the brain, I think it makes sense for us to start thinking more consciously about what foods we should start putting in our stomachs.
Other questions? Just one at the back.
Hi. I was just wondering about time of day, because you hear about young people learning better in the afternoon or evening, whereas they should ban 9 o'clock lectures.
Yes, good question. So the first studies that were done on this were by at the Medical Research Council Laboratory of Molecular Biology, and they took slices of mice brain. So just imagine that if I go up your nose with a pencil, right all the way up through the skull, you'd eventually reach this little kind of really dinky P-shaped structure that's really small. It's about the same size as a pinhead, a very small pinhead, not a drawing pin, but like a dinky little pinhead. There's this dinky little brain region called the suprachiasmatic nuclei that contains around 10,000 nerve cells. And it's involved in helping us regulate our day-night cycle. And other mammals also have this suprachiasmatic nuclei, and it's involved in regulating their day-night cycle. And using these green fluorescent protein kind of microscopy technologies of tagging and dyeing different proteins that are being expressed within the cells, what they did with the researchers was they went into the mouse's brain and took slices of this superchiasmatic nuclei and then imaged the slices. The slices were still alive. They were being bathed in nutrients in a petri dish, and they were still electrically alive, so they were working as functional neural circuits. And the cells had been labeled with this green fluorescent protein that was tagged to little proteins that were associated with directing the day-night cycle. So it's quite complicated, but hopefully that makes sense. And basically what they were seeing was that there's some mice that like to wake up earlier, because they're nocturnal mice, that wake up earlier in the night than other mice. And you could see these waves of electrical activity occurring depending on which cycle, whether they were early risers, the equivalent of human early risers, or late risers. And you can see different waves of temporal kind of resolution of how that occurs in terms of how the genes express the proteins within these nerve cells. And the same thing happens in humans, which explains why there's some night owls and early larks. It's actually written into the way that their genes are expressed and how that's regulated by time of day. Now, adolescence, during the adolescent period, there seems to be some disruption of that, and it's not properly understood exactly how, but probably under hormonal influence. That means that even the early risers of teenagers, the way that their superchiasmatic nuclei directs the rest of their day-night cycle in their brain and how alert they feel basically gets shifted backwards, which is why there's been calls from, particularly, there's lots of research that's going on at Oxford University here with Russell Foster on this. There's been lots of calls for the school day to be put backwards. And in fact, there's been some studies that have been looking at how this does improve learning and memory and alertness within the students for those schools that have participated in these trials. So it does seem biologically, neurobiologically, it makes sense to do this, to not have 8 o'clock in the morning or even 9 o'clock in the morning lecture start times, but to just bump it back a little bit later during the adolescent period.
I'll shout. Science fiction is full of films and things where they download information into the brain, and I don't need to learn anything. It feels like my job would be a lot easier if we could do that. What's your thinking on is that possible? Can we download information into a brain ever? Is that the future of education?
It'd be uploading.
Oh.
So there's been some actually quite scary research on this recently. So they've been, first of all, trying to see whether we can connect to people's minds when they're located in different areas and they're not communicating with each other through different ways. One person was asked to wear an EEG cap, which picked up the electric signals in their brain, and they were asked to think something. And then there was an attempt to transduce that information into an electric signal, which would then be conveyed to another person which was wearing EEG, and that electric signal would actually activate some of their nerve cells so that they would start thinking what that person was thinking without them communicating it. So this was first done 5 years ago, and in fact what the researchers were able to do was place one person in Europe, I don't think it was London, I think maybe it was in France, and the other person 5,000 kilometers away in India, and use this technology to transfer thoughts from one person to another. More recently, there's been some research looking at how we can use this technology to teach an individual something and then take that signature in the brain of that learnt thing and place it in another individual. And what they've found, these are initial studies have been done in mice, and what they found is that the process of doing this actually primes the second mouse's brain so that it learns much quicker. So it doesn't instantly get that information, it doesn't receive it like that, but it is primed to receive the information so it learns a little bit better. And there's also scientists that are working on prosthetic extra hippocampal devices, which are kind of like an external hippocampus memory that might be embedded. So this is going to be the topic of my next book. So I haven't done a huge amount of research on it yet, but it's pretty mind-blowing, isn't it? Yeah. Yeah. It's not yet ready to be applied to the classroom.
For the last thing you said about the external hippocampal is like an external hard drive. Is it what is the future for the treatment of Alzheimer's?
Yeah, so I think that's one of the reasons that the scientists have been looking at it. Because there's an increasingly aging population, and so Alzheimer's is going to be an increasingly financial kind of unmet need unless more advances are being made. But also there's lots of reasons why people would be interested in expanding their memory and being able to more quickly communicate their memories and their perspectives with other people. It's a bit mind-blowing, isn't it? Yeah, I mean, it's not quite there yet, but scientists are looking at it in reality.
Hi, I'm interested in your view as to the effect of digital technology on our brains, and particularly some of our students work for long periods of time, particularly in the gaming technology area. They may work continuously for 1 or 2 days without sleeping. What would be the impact on their brains from your perspective?
Well, as I mentioned earlier, sleep is an incredibly important function because it helps to clear toxins out of the brain. And it's really interestingly, actually, we're talking about Alzheimer's, and one of the overarching themes that reoccurs across quite a lot of neurological and even psychiatric conditions, for example, depression, psychosis, schizophrenia, anxiety, Alzheimer's, for example, is that individuals have long kind of like lengths of interrupted sleep. And that really seems to distort the way that they view the world and how they interact with it. So sleep is important for the ability to remove toxins from the brain, which might attack those connections within the brain, but it also helps to consolidate those little connections that have just formed into mature, stable connections, memory spines. So long periods of not getting enough sleep is going to have quite profound effects on your health, not just your brain health, but it's also implicated in increased incidence of obesity and diabetes, because you're kind of craving certain foods as well that aren't particularly good for you because you're in such a mental state and your brain is instructing you to go for those foods. There's also the effect of the screen. So we've evolved to get signals from specifically the colour blue that will actually help us via this superchiasmatic nuclei region that I was talking about earlier, help us to stay alert and to stay awake. Because when we wake up, we see this blue sky on the savannah, and so that woke us up and primed us for activity during the day. And quite a lot of screens emit a high amount of light that's in the blue wavelength. And there's different ways that you can tweak with the settings of the computer so that you can diminish that or you can get a filter for it. So again, I'd recommend that. In terms of younger children spending a lot of time, or teenage, or just on... Sorry, what was your other question? Part of the question?
My concern is for our students who work for 1 or 2 days at a time, as far as I've heard, and what the big impact might be on their brain structure.
Yeah, so not positive, really. If they do it regularly, yeah, it's not healthy. But then if you do anything continually for 2 days, it's probably not very healthy, is it?
Really? Yeah. So I guess the question I had was concerning the Alzheimer's conversation, like with permanent cognitive impairments or disabilities, whether it's acquired from brain injury or Alzheimer's or whatnot. Are there any things that we can do to facilitate new learning processes within these individuals? What are your thoughts on that?
Yeah, so there's quite a lot of scope for plasticity within the brain, and also redundancy. So again, anything that will help to promote this plasticity, so that any lost functions within the brain could be replaced by other areas of the brain taking over or helping to boost plasticity so that you can recover any lost functions. So basically any of these things would be healthy for anybody. And in fact, antidepressants, one of the modes of their action is to help boost this connectivity, the microtubule dynamics in the brain. So yeah, anything that we can do to help with that is good.
And my questions relate more to the future. So as we're learning, I would not say less content material, but a lot of things is more automated. We're doing much more based on technology nowadays. Do you think the shape and the brain regions are going to change? So the brain of, let's say, in the next 10 years of an adolescent's learning looks much different than it's now, like hippocampus, prefrontal cortex, executive functions. Do you think we're going to see a shift in the shape of the human brain due to the way we're learning and the type of material that we're learning?
Yeah, so there has been a couple of studies that have been looking at the effects of when touch-screen television, telephones first came out, and what they found. Oh no, this was with the mobile phones. This is way back in the day. When mobile phones, you used your thumbs to text. And basically they found that as people were learning to do this and doing this more and more, there was a region of the brain that's involved in controlling their thumb that started expanding. So there's a region of the brain called the motor cortex that's involved in directing movement of your muscles throughout the body. And there's particular areas that are involved in directing your thumb kind of fine motor control movement here. And basically as people were kind of learning to text and doing it more and more and more, these areas expanded. And there's been other studies that have looked at, for example, I mean, the first study that looked at this type of thing, it was the London Black Cabbie driver studies. So as they learnt their way through London and navigated without the use of sat-navs and learnt the knowledge, like the A to Z of London by heart, then a particular region of the hippocampus actually expanded as they learnt the knowledge and as they passed it. But this was at the expense of another region of the hippocampus, because you've got to realize that your brain is, the space of your brain, the volume that it can take up is constrained by the size of your skull, and you can't just kind of like keep exercising different areas and they'll just keep growing, because otherwise it would just explode out of your skull. But since those exciting initial results, what people have found out, or what we seem to be finding out, is that actually during the initial learning phase, there are all these new connections being made. There are these areas that are bulking up in terms of their volume, literally in terms of their volume, as new branches are kind of forming, and new connections being made, new kind of navigational routes being made. But actually, as we refine that learnt skill, that learnt thing into a proper skill, into a refined behavior, then actually the branches kind of form more of a simple, neat structure in the brain, and the volume kind of goes down a little bit. So we might see an initial boost, but then maybe it'll kind of all flatten out.
So I don't know to call an end to the questions there. So I would like it if you would just join with me, please, in thanking Hannah one more time.